All right, we are on to biology. Fun stuff. Um, well, it's been quite a while since the last one I did, which was electronics. So, as a kid, I got. So, this was unfortunate because I was actually good at biology as a kid, but it was actually my worst grade. And I think the reason was because we had such a terrible biology teacher that just wasn't teaching us the stuff. So, um, yeah, it's all thanks to my teacher. Other than, I mean, if it wasn't for my teacher, I probably would have done well. So, um, the question is, will I do better today than I did as a kid? That's difficult to answer because, on the one hand, GCSE biology, there's a lot of very specific GCSE knowledge you need to know. That's just, you just need to know for the exam. For example, the different parts of the body, the different parts of, I don't know, the eye or the penis or whatever. Lots of weirdly named things that you need to know that you'll never remember again. So that's one thing. But on the other hand, I am actually pretty knowledgeable on biology. I do read up on it a lot, and I even worked in I did PhD research in bioinformatics, which is more computer science than biology, but it still involves knowledge of proteins and diseases and stuff like that. So I've no idea whether to predict higher or lower. I've gone with lower, so I think I'm actually going to do worse than I did as a kid for the former reason, because there's a lot of definitions and terminology that I've probably forgotten. All right, well, let's see what happens. Probably won't need to use the writing tablet much. But, okay, let's just use it at least for this. So we got center number 8008. 5. Candidate number 2. X. Yep. Time allowed 1 hour 45. Oh, I forgot to open the timer. I think I'll put the mouse back in. I don't think I'm gonna actually need the writing tablet for this for biology. I'll switch to it if needed. Okay. Where's... I thought I opened Live Split. Where's Live Split? Damn it, there's always one thing I forget to do before the stream. Why would it not open? Live Split. Oh, there we go. Okay, open splits. No, that's fine. Wait, no, it's not visible on hang on. There we go. Okay. Okay, so we got 1 hour 45 minutes. That's quite a lot. I probably won't need that much time. You must have a scientific calculator. Okay, I need to open Google for that. Okay. Use black ink or black ballpoint pen. I'll use dark blue. I'm sure they'll allow that. Pencil should... blah, blah, blah. The maximum mark is 100. The need for good English and clear presentation in your answers. Alright then. Alright. I think we're ready. All right, so Figure 1 shows the student preparing the cells. Prepare some animal cells to view using a microscope. Name 2 pieces of laboratory equipment the student could have used to prepare cells to view using a microscope. Well, I'm looking at this diagram. I think, what is this thing called? Glass slide? I don't know what that's called. Let's call it glass slide. I mean, that's what you would put. What other equipment would you need? That's all you need, isn't it? Or Petri dish, maybe? It's like you don't need to use it, but it might be something you would use. Can I just... Okay. It's just, Is that... I don't know. Prepare cells. Like, I know you put iodine or something on it, but that's not equipment. Alright. Name part A. The lens? Is that what you call it? What is the function of part B? Oh, good, I don't have to name it. To control the focus, right? Also, why have I got so many live splits open? Hang on. Okay. Control the focus. Increase, decrease the magnification focus. Student tried to look at the cells using the microscope. Suggest one reason why the student could not see any cells when looking through part A. Because the magnification wasn't correct. Part B needs to be adjusted accordingly. So he was too zoomed in or too zoomed out, I don't know. Red blood cells are specialized animal cells. Compare the structure of a red blood cell with the structure of a plant cell. Oh my god. Okay, I actually remember some things here. 6 marks though, oh my god. Okay, red blood cell does not have any nucleus, whilst plant cell does? Does it? Maybe? I don't know. But wait, plants have different cells in different places, don't they? So it's not... Blood is a specific type of cell within the organism. Yeah, that's fine. Alright, red blood plant cell does. Okay, that's one difference. Oh, plant cell has a... What's it called? Fibrous. It has a thick outer wall made of... Oh, I forget what the name is. Plant cell has a thick outer wall which red blood cell does not have. What else? That's all I can think of. Oh, this is not going to get me 6 marks. I think plant cells, I think they have rectangular shape, whilst red blood cell is more spherical. Elliptical. Let's try and cover our bases here. I don't know. Is that even true? I don't know. Plants. Structure of a plant cell. What do you get in a cell? You got the nucleus. That's all. That's all I know. If you don't have a nucleus, what else do you have? I don't know. When placed into a beaker of water, a red blood cell bursts. A plant cell does not burst. Why would it burst? I don't know. That doesn't sound very stable. Alright, okay. Osmosis. I think it's osmosis. I think osmosis. When red cell is in water, the water transfers from high concentration to low concentration through the semi-permeable membrane around the cell. I didn't even know this happens, but whatever. When the cell contains too much fluid, the cell will burst. Plant cell, on the other hand, it has that wall. So it's... But what's the name of it? We need to name it. Oh. Oh, it's on the tip of my tongue. It begins with A. A-D. Add something. No. Has a thick wall around the cell, preventing fluid exchange in and out of the cell. I think that makes sense. I didn't know this happens, but okay, now I know. Three different antibiotics. Agar plate. Hang on, is agar plate the thing we need? For the earlier question. I feel like glass slide isn't. isn't correct. But then agar plate. Let's just use it. Agar plate. Okay, bacteria growing on agar. Filter paper disc containing antibiotic A, B, C. Okay. Used aseptic techniques to make sure that only one type of bacterium was growing on the agar. Describe 2 aseptic techniques the student should have used. What? So we want techniques to make sure only one type of bacterium was growing on the agar. I don't know. I can't think of anything even remotely sensible here. How'd you get the bacteria on in the first place? I don't know. I can't answer that one. All right. The student placed the agar plate in an incubator at 25 degrees for 48 hours. Which antibiotic is the least effective? B. B had the smallest range of effectivity. Is that even a word? As bacteria were able to grow closest to the source of the antibiotic. Calculate the area where no bacteria were growing for antibiotic C. Use pi equals 3.14. Give the unit. Huh? What? We've not been given any measurements. Am I supposed to measure this with a ruler? What? Well, I can't do that. Oh, we actually... Foxit PDF Reader actually has a tool for this. So we want to... So it's for C. So we want to measure... Calculate the area where no bacteria were growing for. So we want to measure the distance here. So that's... Damn it. Alright, this is... Oh, for fuck's sake. Okay, can we please have centimeters, please? Okay. Alright. 1.05 centimeters. 1. 1.06. Alright, we'll go with 1.06. Fine. Okay, take this off. So... This is just the outer circle. Circle has radius 1.06 centimeters. So area equals pi times 1.06 squared equals 3.14. 3.14 times 1.06 squared. 3.53. 3.53 centimeters squared. Inner circle has radius oh, you can actually measure area as well. It's probably not what they want you to do though. 0.49, let's go with that. 0.49, right. Damn it. .49 centimeters. So area equals pi times 0.49 squared equals 0.754. Calculate the area where no bacteria were growing for antibiotic C. But wait. It's just the white area. Yeah, it is the white area, right? So we actually have to subtract C itself. I assume so. So area equals 3.53 minus 0.74 equals 3.53 minus 0.74, 2.776, 2.7, let's round it up to 8. Okay. 2.78 centimeters squared. So just one way the student could improve the investigation. Take repeats. Take repeats is always an answer, isn't it? I bet it won't be. Repeat the investigation and take averages. All right, body mass index, BMI. So this one uses the square height, which is supposed to be more accurate, but which is the BMI category of person A in table 1? Person A has BMI 23.1. Hang on, so 63 kilograms. Okay. 1.65 meters in height. 1.65. So normal. So we got 63. And 1.65. Okay, normal. Calculate value X in table 1. Body mass divided by height squared. Okay. 92 divided by 1.71 squared. (92 divided by 1.71 squared equals, and that's 31.46, so round to 31.5. Divided by 1.7. Yeah, that's it. That's easy enough. Scientists think there's a link between BMI and life expectancy. I'm sure there is. Okay. Describe two patterns shown in table 2 about the effects of BMI category. Predicted number of years living in good health after the age of 50. Predicted number of years living in bad health after the age of 50. Two patterns. Well, there are two clear things here, okay. The higher the BMI category, the lower the number of years of good health after 50, and then the exact other way around is the case. The higher the BMI category, the higher the number of years of bad health after 50. Those are two patterns, right? Number of people who are obese in the UK is increasing. Explain the financial impact on the UK economy of the increasing number of people who are obese. Well, UK is free healthcare, so greater strain on the healthcare industry caused by more patients experiencing bad health that's self-explanatory, isn't it? I don't need to say why that would affect the UK economy. This requires more taxpayer money to I don't know. Okay. 2 marks. Do they want us to say something else? Increasing number of people who are obese. What other financial impact? I mean, that is the impact, isn't it? What else? But say more people of severe who are severely obese are unable to work a job and provide input to the economy. More people who are severely and categorized as disabled, unable to. That affects the economy, right? Person who's obese is more at risk of arthritis. Suggest how arthritis could affect a person's lifestyle. Make it more difficult for the person to move, walk, and perform manual tasks. Person who eats a high diet in saturated fat might become obese. Name 2 health conditions that might develop if a person eats a diet high in saturated fat. Do not refer to arthritis in your answer. Alright. Heart disease. What else? What else? Obesity. Oh no, they already said they become obese. So not arthritis. Not diabetes? But that's not really... I don't know. That's more sugar, isn't it? But it's correlated. No. 2 health conditions that might develop if a person eats a high diet. Heart disease is the number 1. It's the biggest killer in the UK. Arthritis. I'm sure I know this, I just can't think of something other than heart disease. Saturated fat. I don't know. I don't actually know. Health conditions. So it could be mental conditions, or I don't think they're counting those. I don't know. I'll just put diabetes. It's probably wrong. All living organisms respire. What is the chemical equation for aerobic respiration? So it's oxygen, carbon dioxide. No. It's got to be... oxygen. And this one is glucose, is it? Produces water and carbon dioxide. I think that looks right. The last one. Definitely uses oxygen on the left and produces carbon dioxide on the right. So that's just... so that's either... Yeah, that's the right one. Name the subcellular structures where aerobic respiration takes place. Mitochondria? I just remember mitochondria. I don't know what mitochondria do, or... I'm just going to say mitochondria. Energy is released in respiration. Give 2 uses of the energy released in respiration. Heat, to keep the organism warm and prevent freezing. And I mean, this doesn't happen for plants, but mussels to allow muscles to contract, allowing the organism to move. Two differences between aerobic and anaerobic respiration in humans. Do not refer to oxygen in your answer. Okay. Anaerobic respiration produces lactic acid as a byproduct. That one everyone knows because that's why you get stitches. Whilst aerobic doesn't. Do not refer to oxygen, because well, oxygen is the most important factor here, but okay, fine. So lactic acid, we mentioned that. And that's a debt that needs to be paid off with more aerobic respiration. So aerobic still uses sugar, doesn't it? Both use sugar. So that's aerobic. What can we say? I don't know. Aerobic respiration causes increased breathing and air intake, but that's not really what they're getting at, is it? I don't know. What are two products of anaerobic respiration in plant cells? In plant cells, I guess they don't make lactic acid. Water, for sure. I think. It's not going to be ethanol. That's bacteria. Anaerobic. Wouldn't it still be lactic acid? It's not going to be ethanol. See, if it's water and carbon dioxide, then that's just aerobic, isn't it? So the two products. Let's have a look at this. So if you were to just cross out the oxygen, just take the glucose itself, what would happen? Well, I think you would not produce the carbon dioxide. So let's say water and lactic acid, because carbon dioxide would use the most oxygen atoms. I don't know. Scientists investigated respiration and photosynthesis using some pondweed and a pond snail. Boiling true, pond water, pond snail. Well lit room for 5 days. After 5 days, cover the boiling tube with black paper. Okay. So concentration of carbon dioxide stayed steady over the 5 days. Wait, so it was always well lit. Okay, fine. Explain why the concentration of carbon dioxide in the tube stayed the same between day 0 and day 5. I think because... Oops. I think they were cancelling each other out. So the snail respired, producing carbon dioxide, whilst the plant photosynthesized, taking in carbon dioxide as an input. The net production, it just happened to balance out perfectly, which is unrealistic, but whatever. The net production of CO2 was 0 as the two processes cancelled out in effects. Why the concentration of carbon dioxide increased? Okay. In the dark, the plant could no longer photosynthesize. Therefore, CO2 was no longer removed. That's it, right? On day 10, the pond snail died. Oh, this is cruel. Explain why the death of the pond snail caused the concentration of carbon dioxide to increase after day 10. The snail's respiration alone caused CO2 to go up. Okay. So when the snail died, the snail's death halted the respiration process. Caused the concentration of carbon dioxide to increase after day 10. Hang on. Okay, it was always increasing. What? So the plant would respire as well, but that's not important here. Caused the concentration of carbon dioxide. Why did it cause? Okay. Okay, well, no, it's not nothing to do with respiration. Is it the snail started decomposing? The dead snail began to decompose. Bacteria growing on the snail's body produce respired and produced CO2. But so did the plants, but isn't that it? Amylase is an enzyme that breaks down starch. Amylase is a polymer of smaller molecules. Name the type of smaller molecule. I don't know. Enzyme breaks down starch. Name the type of smaller molecule. I have no idea. Protein? No, that's not. It's the best thing I can think of. Protein. Whatever. Name the 3 parts of the human digestive system that produce amylase. I'll just name 3 parts of the human digestive system because I don't know. Stomach. Small intestine. Large intestine? I am not going to do well at this at all. Explain how amylase breaks down starch. Answer in terms of the lock and key theory. The what? Okay. Lock and key theory helps me here. I reckon, I don't actually know, but I reckon amylase molecule, amylase enzyme has a particular shape akin to the key in the lock and key theory. This shape can combine with the shape of the starch particle. So starch is the lock in this analogy. When the 2 fit together, I'm just making this up, but it sounds plausible. When the 2 fit together, then the enzyme can perform the breakdown process. I'm just getting that from lock and key theory. Alright, be right back. Effective temperature on the activity of amylase. It's got water at 5 degrees, we've got starch, we've got amylase. Excuse me. Alright. After 5 minutes, pour the starch solution into the amylase solution and mix. Remove 1 drop. Place it on a spotting tile. Immediately add 2 drops of iodine. Okay. Name 2 control variables the student used in the investigation. 2 control variables. What's a control? Oh, they've changed them. They used to, in my day, they were called dependent and independent variables. Now it's control and something. Right, control is something you control, right? So temperature. Just time? Is that the variable here? Time. Because you're doing it every 5 minutes or whatever. Why did the student leave the starch solution and amylase solution for 5 minutes before mixing them? To allow the products to reach an equilibrium in temperature with the water surroundings, just to reach temperature equilibrium. What conclusion can be made about the effect of temperature on amylase activity? What does yellow-brown even mean when... ...20 and 65. Okay, between 20 and 65. It did not become yellow-brown. 5 minutes it took. So yellow-brown... So the longer it takes... So 2 means the activity was the highest. Activity was at its peak around 35 degrees C. If the temperature is decreased or increased from 35, then the rate of activity decreases. Explain the results at 5 and at 80 degrees. Use table 3. I mean, it just means 5 marks. What can I say here for 5 marks? It just means it wasn't active, right? Amylase was inactive at these temperatures. Well, that's all you can say. Why am I supposed to get 5 marks from this? What? Do I say something? Okay, let's make up something about the shape or so. Okay. At these temperatures alter the shape of the enzyme to the point that they no longer fit with the starch, the lock and key process breaks down. Just making that up, but still 5 marks. What the heck? Skip it. The student investigated the effect of temperature on amylase activity. Yeah, that's what we did. Okay. Describe how the student could extend the investigation to determine the effect of a different factor on amylase activity. What factor? Tell me the factor. What? Just change that other factor. What? Keep the temperature fixed at... Ooh, I would want it at 35, right? Just 35 degrees C. Repeating the process, but this time only altering the new factor will allow us to investigate this new factor. Record the time taken for iodine in the same way as before to determine the amount of amylase activity. I don't know. Figure 9 shows a cross section of a leaf. Which cell is most transparent? Huh? What? They're all transparent. What? Okay, D is quite small, C is bigger, B is... What? I'll go with C then? I don't understand this question. Which cell structure in a leaf mesophyll cell is not found in a root hair cell? Root hair cell, like in a mammal? Which cell structure? Well, the outer wall, but I don't know what it's called. I don't know. Plants lose water through their leaves. Name the cells in a leaf that control the rate of water loss. Oh my god. No, I don't know. Water is taken in by the roots. Oh no, root hair cell is the plant root. What cell structure? Oh, roots don't photosynthesize, so... But that's not... Chlorophyll is a chemical, it's not a cell structure. Whatever, just say chlorophyll. Name the cells in a leaf that control the rate of water loss. Don't know. Water is taken in by the roots, transported up the plant, and lost from the leaves. Which scientific term describes this movement of water? Oh. Oh, it's on the tip of my tongue. Oh. It's not phototropism, that's something else. But I think it is something to do with tropism. Hydro is water, so would it be hydrotropism? No, because I think tropism is to do with the way the plant grows. Not, just it's better than nothing. Which change would decrease the rate of water from plants' leaves? Wait. What's stomata? Alright, I'm adding stomata to this question. I don't know. Stomata. Alright. I think temperature, right? No. So why would the plant prevent water loss? Because it needs the water. But it always needs the water. But temperature would... I think it's just, I don't know, temperature. Compare, oh no, compare the structure and function of xylem tissue and phloem tissue. Oh, I don't know what either of those things are. I think it's like xylem is like this hard stuff. Xylem tissue is harder and used, is this to support the plant's growth, whilst phloem is softer and used for transporting materials through the plant? I have no idea. If that's true, then xylem has, I don't know, thicker cellular walls which can be controlled in thickness in order to direct the plant to grow towards the sun. I'm bullshitting so much at this point. And other factors. I don't think that's right. I think xylem is maybe for water and I don't know. Figure 10 shows the total volume of water lost from a plant over 6 hours. Okay. Determine the rate of water loss at 12. Use the tangent. Okay, so that's at 12. So the gradient. Okay, so we got since 230, and then I got 190. So between them, that's 40. And then here we've got So 7 hours. Okay, so delta y equals, say, 40 centimeters, 40 centimeters cubed. Delta x equals 7 hours equals 420 minutes. So gradient equals 40 divided by 420 equals 0.0952 cm cubed per min. That's standard. That's not standard form. So 9.52 times 10 to the minus 2. The rate of water loss at midnight was much lower than at 12. Well, midnight is not on this chart. What? Okay, they're just telling us. Because it respires, so at night the plant stops photosynthesizing and begins respiration, which produces water as... Yeah, but this doesn't... That doesn't tell us why water loss is lower. If anything, that would cause water loss to be higher. Why does the plant even lose water? I don't know. I don't know what the plant uses the water for. You would think it would be losing more during the nighttime, according to that logic, which I just tried to write. I don't understand plants. So that's not useful. What? Is it temperature? I think it... What did I take earlier? Increased temperature would decrease the rate of water loss. That's what I claimed. But it's not warmer during the night. Oh, for goodness sake. I don't know. Skip it. Let's just leave what I wrote there. But that doesn't answer the question. Figure 11 shows where 3 of the same type of tumor were found in a patient. Okay. So that's the lung. And no, that's not the lung. Is it? Yeah, that's the lung. That's the liver. Describe what happens to cells when a tumor forms. They replicate, right? Cells replicate. What evidence is there in figure 11 to suggest that the tumor in the lung is malignant? It's bigger. It is bigger, right? Some types of cancer can cause the numbers of blood components in a person's body to fall to a dangerously low level. A person with one of these types of cancer may experience symptoms such as tiredness, frequent infections, bleeding that will not stop after the skin is cut. Explain how a very low number of blood components in the body can cause these symptoms. Oh no, I don't know. Okay. Let's reason this out. Blood contains platelets, which can be used to clot and prevent bleeding. Low blood components would result in fewer platelets, which makes it harder to stop bleeding. Okay, that's something. Frequent infections. Blood contains white blood cells to fight infections. With lower blood components, it would be harder for the person's blood to fight off infections, so they will have a stronger effect. Tiredness. Why would what tiredness? I don't know. Low blood pressure. What is that even? Okay, this probably doesn't make any sense. Low blood components causes low blood pressure, which causes feelings of fatigue or tiredness. I think everything in that sentence is probably wrong. Did we really learn this in school? Some patients with a very low number of blood cells may be given a blood transfusion. Blood transfusion is where a patient receives blood from a donor. Different people have different blood groups. The red blood cells found in people with different blood groups, okay? The antibodies that could be made by people with different blood groups. Yeah, okay. Antibodies can bind to antigens that have complementary shapes. Yeah. When antibodies bind to the antigens on red blood cells, many red blood cells begin to clump together. Each red blood cell is about 8 micrometers in diameter. Many capillaries have an internal diameter of about 10 micrometers. Okay, what are you going to ask us? In one type of blood transfusion, only red blood cells from a donor are transferred to the patient. It is dangerous for a patient with blood group A to receive red blood cells from a donor with blood group B. Okay. Okay, well, B has type B antigens, which would bind to the antibodies anti-B in A's blood. This would cause the donated blood to clump. Okay, why is that dangerous? What does that mean? Could it cause a blood clot? I don't know. Clump. This could cause a blood clot, which could be deadly. I don't know. Could it? Probably. Explain why blood group O red blood cells can be given to patients with any blood group. O's red blood cells will not bind with any antibodies, so no clumping will occur with the donated blood. That's it, right? Table 4 shows some of the risks associated with blood transfusions. Allergic reaction, hepatitis B, hepatitis C, and kidney damage. Which risk has the lowest probability of occurring? One in... So... Oh, they're just giving these numbers in obscure... Wait, so 1 in 3 times 10 to... Wait, so that 3 times 10 to the 5 is in brackets, so let's just 1 in 3 times 10 to the 5. Wait, 3 times 10 to the 5, so 1, 2, 3, 4, 5. Okay, so that's what it looks like. So it looks like this, because that's just a weird way for them to write it. 6.7 times 10 to the minus 7 would be smaller. And 1 in 70,000 would be... that. Let's write this down. And 6.7 times 10 to the minus 7 would be much smaller than either of them. Okay, so it's hepatitis C infection. It's just a weird question. A person has a tumor blocking the tube leading from the gallbladder to the small intestine. Explain why this person would have difficulty digesting fat. gallbladder secretes. Oh, what's it called?
I love you.
The thing that breaks down fat. I don't remember the name. An enzyme which emulsifies? Is that the right verb to use here? Is it even an enzyme? I don't know. Fats. The blockage would prevent the... Is it even an enzyme? From reaching the small intestine where food is being produced, hence fats wouldn't get broken down. Why is it 5 marks? Oh, I've done terribly. I'm never gonna be a doctor. We've got just over 20 minutes left, but I don't even want to go back and do anything. I just want to end it there. Oh, it was horrible. I just inhaled a fly. 1 second. All right, well, I don't feel good about that at all. Maybe I was right to have a poor prediction. I think I'm gonna even do even worse than my prediction though. It's like the questions don't seem hard, but it's this subject is really something you have to study. I can't just bullshit my way through like I could for physics and stuff. Like you actually have to know the terminology. It's knowledge. It's more knowledge than skill. Whereas maths and physics and electronics, I don't know about electronics, but it's kind of more. There is a lot of skill involved using your smart senses. But here, like, what the heck, xylem and phloem? I don't know. I can go back and answer some questions using common sense, but I'm just too depressed now. Let's just leave it there. All right, let's mark this, get this over and done with. All right. Excuse me. Okay. All right. Oh no, this first question is already gonna be. Yeah. Okay. So, any two from microscope slide, cover slip, dye stain. That's not equipment. Mounted needle, pipette dropper, scalpel, forceps tweezers. I got none of those. 0. I'm gonna have to start writing these down. Alright. 2 marks lost, so just count the marks I've lost. But that's what I usually do, but in this case, probably actually want to count the marks gained. Okay, we'll just carry on. Lens. Is it called a lens? Yes. Increase, decrease the magnification focus. Yes, to focus. Yeah. Do not accept reference to magnification. But that is literally what it does, right? But I did say to focus. So they're saying ignore anything where they're talking about magnification. But I think it's still right, as long as I talked about the focus. That's weird, but usually it's on a positive marking scheme, so I believe that would still get the mark. Magnification wasn't correct. Okay, I shouldn't have mentioned magnification there, but... Allow idea of magnification not being high enough. Okay, but anyway, I think I got that right. Part B needs to be adjusted accordingly, so I was saying the magnification wasn't correct. Yeah, they said allow idea of magnification not being high enough, so I think that's fine. Okay, 6 marks. Alright, let's have a look. Red blood cell has no nucleus, plant cell has nucleus. Okay, correct. No cell wall. Why didn't I say cell wall? Why did I say thick outer wall? Sort of right, I suppose. Plant cells have a rectangular shape. Is that even a... Vacuole, permanent vacuole. Smaller. Cytoplasm, cell membrane, pigments. Similarities. I didn't mention any similarities. So basically, I mentioned how many things do you need to mention though? I think I got level 1. And what I said here is probably can be correct, but not 100% correct because plant cells can be different shapes. But it is sort of correct. Spherical, elliptical. Yeah, that's kind of a disc, but then again. So that's correct, but not important, so we can ignore this. It's not wrong. Maybe I get 3 then, I don't know. I mean, I did mention a few differences, it's just... I don't know, 3. That means I lost 3 marks. Okay, osmosis. Water enters the cells by osmosis. Plant cell has a cell wall, prevents boasting. Yeah, I got that. Oh, this I did not even write anything down for. Let's have a look. Sterilize equipment before use. Well, that's obvious. Why didn't I say that? I would have got that 1 mark if I had written it down. Alright, well, 0 marks there. B. Kills the smallest area. Yeah, that's fine. 2.78. .1 centimeters. What? So I got... Okay, first of all, the 1.1, I don't have this on paper printed on A4, so I can't actually know. So the only thing we can do is pretend I got it right, got the distance correct, the radius. So I said the radius was 1.06. Okay. They say it's actually 1.1, but we'll pretend I had 1.1, because what else can I do? My calculation with that was correct. So that is the answer. But I didn't subtract. But you're not supposed to subtract this circle here? Well, that's not clear from this diagram, because look, area where no bacteria are growing is pointing directly to the white space, not the shaded space. So I have the right answer there, but I didn't put it in the final space. So how many marks would I get for that? And that's so unfair, because I would say from this, it's telling us to subtract the shaded area and use only the white area. But apparently the shaded area is also included. And I can sort of see that because it says the area where no bacteria. So of course that should include the shaded region, but it's still not clear. All right, so I had the right answer, but then I did a subtraction that wasn't needed. So I mean, I would say that's 4 marks because the right answer is right there, but I just did something extra. So I would think 4. It would either be 3 or 4, I think. But I think 4, because if the right answer is actually staring at me right there, then that's good enough. And when I say right answer, I mean right answer assuming I have this on paper with a ruler. It's just one way the student could improve the investigation. Repeat the investigation? Yes. Okay, normal. Correct. Alright, I'm actually not doing too badly. 31.5, correct. Higher the BMI, lower number of years, good health. Higher BMI, higher number of... Yeah, I mean, straightforward. Costs the health service more money, yep. More time off. Allow more people unable to work. Yes, I did say that. Okay. Make it more difficult for the person to move. Yep. Heart disease and diabetes. So heart disease, yes. The others, no. Oh, type... I didn't say type 2 diabetes. I said diabetes. They've underlined type 2, so I think you needed to say type 2. Allow cancer, allow liver disease, high blood pressure, high blood cholesterol. Okay, there were lots of options I didn't think of. But I did not say type 2. All right. So it's that one. Yeah, that's correct. Mitochondria. Yes. Remember that one. Heat. Keep warm. Movement muscle contraction. Yep. Produces lactic acid. Yes. Correct. Aerobic respiration causes increased breathing. No. So 2 marks. I mean 1 mark. Breathing is not. That's nothing. That's not like a consequence. It's just. What are the 2 products of anaerobic respiration in plant cells? Lactic acid and water. Fuck. Carbon dioxide and ethanol. God, that's so wrong. But is that? Don't know. I did not know that. Okay. All right. So yeah, the pondweed is not respiring because it's not dark. So why is it saying snail and pondweed? I know that plants respire, but they respire when it's dark, don't they? So what? Either way, I think I've correctly answered the question. I did say pondweed. So, yeah, I did mention photosynthesis, so yeah, I think that's correct. No photosynthesis. Okay, yeah. Snail is being decayed to be bacteria, therefore respiration releases CO2. Yes, perfect. Protein, no, it's amino acid. Name the 3 parts of the human digestive system. Oh god. Oh, it's not going to be these. It's going to be... Small intestine was 1. Yeah, okay. How many do I get? 2 correct for 1 mark. So I got 0. Starch binds to active site of enzyme. What? Because shape of active site are complementary. I think I got 1 mark for saying they fit together. I didn't mention active site though. I think 1 mark then. Temperature and time. 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 Ignore time unqualified. Do not accept temperature. Why? Balls. But you are controlling the temperature. I don't understand this new control variable system. We used to call it dependent and independent. And I'm pretty sure those would be dependent variables. No, independent. Oh. Allow reach the same temperature, correct. Alright, I basically said that. Just kind of in two directions. Explain the results of 5 and 80. So I've sort of said the bit about the shape. So the starch can no longer fit. Yeah, look, I literally said no longer fit with the starch. But this applies to 80 and not 5. So how many marks? Oh, that's a difficult one. If we ignore what I said about 5 and just look at the 80, I think I got those 2 marks. The fact that it doesn't hold for 5 is irrelevant. That just means I didn't get the 3 marks for that one. Okay, so I think 2 marks because I got the description of 80 correct. Keep temperature constant, but change named factor and test a range of values of named factor. Sure. That's what I said. Which cell is most transparent? Don't understand that question. A. Nope. Why is it A? It says something in this diagram that you would see on paper that you don't see here, because I don't see what makes it transparent. I think it's just the fact that it's the cell wall, the plant wall cell. Maybe it's just known that that's the most transparent. I don't know why, but... Okay, that's wrong as well. Alright, so we got... Stomata? Nope. Ignore stomata. I don't even know what stomata are, but I just copied it from something I saw later. Wrong. Yeah, hydrotropism is about where the roots grow towards the water. It's nothing to do with movement of water. So... increase temperature. Wrong. Oh god. This one. Alright. Xylem is made of dead cells. Phloem is made of living cells. Xylem is hollow. Cytoplasm. Oh my god. So wrong. I sort of remember that now. Xylem transports water. I think I even mentioned that, but I just didn't really want to go with that. Yeah, that is a big fat 0. That is a loss right there. Oh my god. Okay. Alright, please. 9.52 times 10 to the minus 2. Correct. Less water loss at night. Stomata completely closed. Cooler. I don't think I got any marks for that. I don't understand why lights would even be relevant here. I don't know. I don't think they've answered the question in their mark scheme. I don't understand why cooler or less light would mean less water loss. I don't see the connection. Cells replicate. Yep, that's correct. Bigger. Metastasized. Wait, what? Oh. It's because the fact that there are other tumors means it's probably spread and caused them. Ah. Okay, well, I didn't get that. I thought it was just someone who happened to have a bunch of tumors that are unrelated to each other. Okay, so tiredness. Yeah, I did not get that. All right, frequent infections, fewer white blood cells, so fewer antibodies produced or less phagocytosis, so fewer pathogens. Sort of said that. All right, bleeding, fewer platelets, so blood does not clot as easy. Okay. I said I got bleeding correct. I think I hit level 2. I got bleeding correct, and I got infections correct, but not in full scientific detail. But the conclusion was correct. So I would say... It doesn't really tell you how to distribute these marks. Like, I would say 3... Okay, level 2, so 3 or 4. Is it 3 or is it 4? I think 3, because I did not get the tiredness thing correct at all, so... I would say 3. Maybe I'm being overly harsh on myself for this one, I don't know. Alright. Anti-B antibodies in patient will bind to type B. So red blood cells clump together. Oh, I didn't... Oh... I got as far as saying clump, but the problem is they block the capillaries, not blood clot. I don't even know if blood clot is correct. So cells have reduced supply of oxygen. Okay, so I got it. I got the first 2. I got 2 points there, basically. 2, no. Yeah, I would say 2 of the points were kind of illustrated here. Yeah, that's correct. Hepatitis C, correct. It's bile. Bile is the stuff that breaks down the fats, so it's not an enzyme, I don't think. I did use the word emulsification, so I got 1 mark. Oh, no, lipase is an enzyme, isn't it? I got 1 mark there. Oh, I see. So the bile would emulsify the fat, but that doesn't mean break it down. So emulsifying breaks it into smaller pieces, which makes it easier for lipase to act on it. That's what they're saying here. So yeah, I got 1 mark, but I did not really get anything good there. That is... Okay, there we go. So I lost 44 marks altogether. Not terrible, actually. So I got 56. Not a disaster. More than half the marks, so I mean, I don't know what would be the, let's see. AQA past grade boundaries. Let's see. For GCSE. For November. So it's what date is this? It doesn't say the date. June. It's June 2021. I thought I was using November. Okay, June 2021. All right, I think this is June 2021. I'm not sure. Okay, let's have a look at this anyway. No, that's... I don't know how to even find the grade boundaries. How did I do it before? Alright, don't worry about it, we'll figure it out next time. Okay, well, not terrible. So, paper 2 be next time. I hope it covers more to do with the human body. I think I know that stuff better. I don't really remember stuff about plants and enzymes and stuff, but give me a diagram of the penis. I can label all the parts easily. Or vagina. All right. Actually, I don't think I could label a vagina very accurately.